Again, we have to learn about the blood platelets. They are the third kind of uh, blood cell. We learned about the erythrocytes, leukocytes. Then another kind is the blood platelets or thrombocytes. Another name is thrombocyte. Then its main function is the clotting of the blood. Then they are the smallest of the blood cell. Then they are produced in the bone marrow at particular cells known as megakaryocytes. A particular large cells are present in the bone marrow. By budding from that megakaryocytes, they are produced. Then its lifespan is 3 to 5 days. We know that this is the 120 days is the lifespan of RBC. 1 or 2 week is the lifespan of WBC. Then the 3 to 5 days is the lifespan of platelets. Then it is destroyed in the spleen. Spleen is the lymphatic organ. Then it at the time of injury or when it rub through the surfaces inside the blood vessels, these plate will disintegrate or destroyed, and it produce a particular substance known as thrombokinase. It is really an enzyme that initiate the blood clotting. Then again, blood clotting is also known as the coagulation. Blood coagulation, we can tell like that. Then firstly, we learned that it produce an enzyme like substance known as thrombokinase. It act like an enzyme, so that it got the kinase. That name indicate the name resemble the name of an enzyme. It is also called thromboplastin. Thromboplast is the cell. It produce the substance thromboplastin or thrombokinase. Act like an enzyme. Then again, this thrombokinase act as an enzyme, and with the help of calcium ions present in the blood plasma. We know that our blood contains calcium inside it. A particular amount of calcium is present inside the blood. This thrombokinase act or react with the calcium ions present in the plasma, and that it convert a substance prothrombin in the plasma into thrombin. Prothrombin is also present in the blood. It's a protein that is present in the plasma, but it is inactive. It is also a protein that is present in the plasma. With the help of calcium ions, it converts substance prothrombin into thrombin. Then that vitamin K that is essential for the production of prothrombin. We have learned that vitamin K is essential for blood clotting. How it help? It help to produce the prothrombin. Prothrombin is a protein that is present in the blood plasma. So prothrombin, thrombokinase with the help of calcium ions convert prothrombin into thrombin. Thrombin is an active substance. Then thrombin, in the presence of thrombin, again calcium ions have two roles. Once it act with the thrombokinase. Then secondly it act with the thrombin in the presence of calcium ions with the soluble fibrinogen. Fibrinogen is also present in the plasma. That is prothrombin, fibrinogen, etc. are present in the blood plasma. Fibrinogen of plasma convert into fibrin. Fibrin is a substance that form the thread. That is an insoluble substance. Form thread like insoluble thread. Fibrinogen. fibrinogen is the substance from which the fibrin is formed. Then that form the sticky threads and that form a network at the mouth of the wound. Then blood cells are trapped in that network of fibrin. Then that form a blood clot, and then it after uh, uh, trapping the RBC in this mesh of the fibrin, then it squeeze out the rest of the plasma, that is the clear liquid known as the serum. The serum is the liquid from which the fibrinogen is removed. You learnt the definition of serum in previous class. So that how the clotting occur. First is the thrombokinase, that is produced by the platelets. The thrombokinase act like an enzyme. It act with the calcium ions that convert prothrombin into thrombin. Then thrombin again act with the calcium ions that convert fibrinogen into fibrin. Fibrin is thread-like that form a mesh-like structure at the mouth of the wound, and RBC get trapped at that area so that it close the mouth of the wound. After some time, that clotted part will shrink and squeeze out the liquid present in it. That coming out liquid is known as the serum. That is how a clot formed at the mouth of the wound. Then again, usually at the time of an injury, this clotting, when blood come out from a wound, the clotting will occur. But if we keep the blood for some time in a test tube, a clot again can be formed. Here it is structure. When some blood is, if we keep it in a test tube, first we can see the fibrin formed. Then on the right side you can see the serum is separated out from the clot. Here again another thing you have to notice that the clot can be formed. Clot can caused by the movement of blood over rough surface as on a cholesterol deposit on inside the blood vessel. I drew a diagram to make you understand here. You can see a narrow blood vessel. Inside it we can see the cholesterol deposited. That through that area, when blood happen to move, these small platelets may happen to rub on that surface, that narrow surface. When it pass through it, it happen to rub against the cholesterol deposit. Then again it happen to disintegrate so that it may produce the enzyme thrombokinase, and in that way a clot may form inside the blood vessel that may cause the uh, blocking of the blood vessels, so that it can cause the heart attack or such things may happen to the person if they have cholesterol inside their blood vessels. Then another important term you have to learn is the hemophilia. Hemophilia is a genetic disorder. Hemophilia, that word you saw in the chapter genetics. Hemophilia is a genetic disorder in which the blood does not clot properly due to the lack of blood clotting protein. You know that in hemophilic person the blood cannot clot properly. In the case of the dengue fever also the same thing happen when blood cannot clot because of the absence of certain enzymes, in the absence of certain proteins, blood cannot clot. In vitamin K deficiency also the same thing may happen. You know that vitamin K is essential for to produce the substance prothrombin, so that the same case may happen to the hemophilic person, that protein cannot be produced. In case of dengue fever, the platelets, the blood platelets may decreased because of the infection. The blood platelets may decrease so much so that blood cannot clot through, even through the capillaries blood may come out in severe cases of dengue fever. Capillaries may break and blood can come out from it. Then again we have to learn about the blood transfusion. At the time of accidents or at the time of operations, sometimes a person lose a lot of blood. In such cases, the person may need blood from another person. So receiving blood from one donor to a recipient, that process is known as blood transfusion. Look here. So when blood transfused, a matching should be there. A compatibility must be there from to both person. The receiving or recipient and donor's blood should match, because several proteins are present inside the blood. We know that. So that a matching is necessary. In so according to the compatibility, the blood is grouped by a person called Lane Steiner. Blood grouping was discovered by Carl Lane Steiner. In RBCs of human being, they have specific proteins that are antigens are present on it. Here I drew certain structures here. One RBC with A group, they have antigen A on its surface area. I drew one figure here. Blood group A persons have their RBC have antigen A on their surface, and in their plasma they will have antibody B. If it is a person's blood with A group, their RBC have antigen A on its surface. Antigen A meaning a kind of protein that is present on the surface of the blood cell. Antigen A, they will have antibody B in their blood plasma. So if the person is with the B group, their RBC have B on its surface and antibody A will be present in the plasma. Antibody A meaning if a group person, if an A group person get the B group blood, that may happen to take some reactions. Antibody B, antibody A persons have antibody B. Antibody B എന്ന് വെച്ചാൽ ബിക്ക് എതിരായിട്ടുള്ളൊരു വസ്തു ആ ബ്ലഡിനകത്ത് പ്ലാസ്മയ്ക്കകത്ത് കാണും. എ ഗ്രൂപ്പിലുള്ള ആൾക്ക് അയാളുടെ പ്ലാസ്മയിൽ ബിക്കെതിരെ ആൻറ്റിബഡി മീനിങ് ആൻറ്റിബഡി ബിക്കെതിരായിട്ടുള്ളൊരു പ്രോട്ടീൻ വിൽ ബി പ്രസൻറ്റ് ഇൻ ദ എ ഗ്രൂപ്പ് person's blood. So a clotting or coagulation may occur if we receive the blood from another group. If it is not matching, a person can take blood from A group or O group blood. Here we are learning about the blood group. 4 kinds of blood group according to the ABO system. There are 4 kinds of blood, that are A group, B group, AB and O. 4 kinds of blood groups are there. A group person have antigen A and antibody B, and A group person has antigen A in his blood, antibody B in his blood plasma. B group has antigen B and antibody A will be there, and AB person have antigen A and B but no antibody A and B. because he does not have and no antibody A and B antibody is not present for the AB group, he can receive blood from any group. So he is known as universal recipient because there will not be any reaction. If he receive blood from any group, he will not have any chemical reaction in his blood because there is no antibody in the AB group person. Then O group person. O group meaning no antigen, 0. You can understand it that 0. No antigen on his RBC, but he will have antibody A and B. So O group person can receive blood only from the O group person, but he can he does not have no antigen. He can donate his blood to any other group. So he is also known as universal donor. O group person is known as O group. O group is universal donor and AB group person is known as universal recipient. Here again you can see how universal donor, here a diagram is there. O group person can give blood to A, B and AB. Here universal recipient, that AB group can receive from O, A and B group. Here also, here a matching blood group of donor, blood group of recipient, a chart is given. A group can give to A group and AB group. Blood group donor, recipient A and AB can receive blood from A group. B can receive blood from B and AB. AB person can AB person recipient, universal recipient, so that AB person can receive blood from any other universal group of the donor. AB can give only to AB person, then O group can give blood to all other groups. Here again another system also you can see, RH system. RH system meaning RH is the factor stand for rhesus or common monkey, that invented, that particular protein first invented inside the blood of, in the blood of common monkey. So it rhesus monkey. So that factor, that particular thing is known as RH factor. That RH factor, when blood of such individual, if RH factor is present in the blood of an individual, that individual is known as RH positive, and if that factor is absent in particular blood group, that person is known as RH negative. So RH factor also has certain important. Here you can see if the RH factor, RH person, if a recipient receive blood from an RH positive blood, that recipient that get sensitized or sense that RH factor and produce an antibody. After 2 or 3 weeks, his blood or his or her blood may produce an antibody after 2 or 3 weeks. Then that may be present or persist in his or her blood. Then if it happen second time, if he again another blood transfusion occur, that cause antibody may persist in the recipient blood, so that a chemical reaction takes place, so that a coagulation may occur that may lead to the death of the person. In case of pregnancy also, it has certain important. If Rh negative woman may become sensitive if she carries an Rh positive child in her uterus. When the husband is Rh positive, the first Rh positive child will be normal. Then in the second time, uh, that may be that blood at the time of delivery, sometimes blood vessels may break and mother may sensitize the blood of the first child. The antibody may form inside the blood of the mother and they may persist. If she consumes the second child with the positive blood, Rh positive blood, the antibody may react from the mother's blood. That may happen to the death of the person or the abortion. Today I am finishing the lesson. Thank you for listening the class. You may try to answer the progress check or the review questions. Then after that I shall also send the right answers for you. Thank you.